Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Welcome to your channel, The Anatomy Canvas, with your tutor, Dr. Shehla Aman. In today's class, we are going to start with the embryology of the nervous system. So the learning objective for the today's class are discuss the development of the spinal cord and development of alar and basal plates and its derivatives, and discuss the congenital anomalies associated with the spinal cord. So first of all, have a look on the cells of the origin of the central nervous system and special sense organs. There are generally three sources from which whole of the central nervous system and special sense organs originate. The first source is the neural plate. This neural plate forms from the epiblast by the process of the gastrulation, where ectoderm is formed, and this ectoderm will form neural ectoderm, which will form the neural plate. So this neural plate will further form the central nervous system, the somatic motor nerves, and the preganglionic autonomic nerves. So this is the first cells which first source from which the CNS arises. Second source is the neural crest cells, which remove themselves by epithelial to mesenchymal transition from the neural plate and migrate away just prior to its function into neural tube. So this second source, the neural crest cells, will form the neurons and the glia of the peripheral nervous system, that is the somatic sensory nerves, the somatic and autonomic ganglia, postganglionic autonomic nerves, and the suprarenal and the chromaffin cells. So this one is the second source. And third source for the development of CNS and the special sense organ is the ectodermal placodes. These placodes contribute cells to cranial sensory neurons and form the olfactory epithelia as well as epithelia of the inner ear, and by a non-neuronal contribution also form lens of the eye. So remember, dear student, each and every line of this slide are your MCQs. So these derivatives are very much important from MCQs of point of view. So please memorize these. So now have a look on this diagram. This is just a revision from your previous knowledge. Here in this diagram you can see this is the neuroectoderm, which is forming from the ectoderm. The cells elongate and they form the neural ectoderm. This neuroectoderm forms then the neural folds and neural grooves, and then when these neural folds fuses in the midline, they will form a closed tube called the neural tube. Before the closure is complete, cells at the edges of these folds dissociate and they show from epithelial to mesenchymal transition, and they form the neural crest cells. So these are this was just a revision slide from your previous knowledge. Have a look on the slide as well, that the nervous system develops from neural plate. Neural plate is a thickened area of embryonic ectoderm. Neural plate then differentiates to form the neural tube, neural folds, and neural crest cells. So neural tube differentiate into the CNS, brain, and spinal cord, and neural crest cell will give rise to cells that form peripheral and autonomic nervous system. Also the third source, which I have told you in the previous slide, that is the placodes, which are the sensory placodes which are forming sensory organs in these regions as well. So neurulation is the formation of the neural tube. It begins in the region of 4th to 6th somite, and in the region of from the region of the 4th somite to cranially, that is the cranial 2/3 of the neural plate, it will form the future brain. And behind this area caudal to this 4th somite, all of the neural tube will form the spinal cord, and the closure starts in the region of the 5th somite usually, and then it proceeds both in cranial and caudal direction till a small pore remains, that is caudal and cranial neuropore. So the neural canal is the lumen of the neural tube communicate with amniotic fluid through cranial and caudal neuropores. The walls of the neural tube thickens to form the brain and the spinal cord, and neural canal is converted into ventricular system of brain and central canal of the spinal cord. Now we discuss all of these things in detail. First, have a look on this diagram. Here you can see these are the pairs of the somites which are present, and in the center you can see neural tube. So in the region of the fifth somite, initially the fusion of the neural force starts, and it proceeds both in cranial direction and in caudal direction, and the cranial neuropore closes at day 25 and caudal neuropore closes at day 27. It is just 2 days behind that of the cranial one. So in this diagram also you can see this is the rostral or cranial neuropore. When it is open, it communicates with the amniotic cavity. Similarly, here is the caudal neuropore. In this diagram you can see the lens placode, the otic pit, the pharyngeal arches, the area where the heart is forming, the septum transversum. This area is the upper limb bud, and all these are different features at this stage in which the neural tube is developing. So in this diagram again you can see this is the neural tube, and in this neural tube you can see this is the area which will form the forebrain. Here you can see the optic vesicle. Then this is the area which will form the midbrain. This is the midbrain flexure, and then the area forming the hindbrain, and this whole will form the spinal cord, where you can see the spinal ganglion as well. So this one is the section taken at this level, and in this diagram you can see this is the neural tube, and on each side you can see the spinal ganglion which are forming from the neural crest cells, then the somites which are present, the notochord, the aorta, and the midgut which is developing over here. In this diagram also you can see different parts of the brain which are developing, that is the telencephalon, also called the forebrain, the diencephalon, the mesencephalon, metencephalon. Here is the pontine flexure and the myelencephalon, and then is the primordial spinal cord. So first have a look on this diagram, that this is the section taken through the wall of the closed neural tube, and in this slide you can see this area is called the ventricular surface. This is actually the area where all of the mitotic figures are present. Here cells divide and then move to the next area which is called the marginal zone. So in this diagram you can say this area is the marginal zone. In between the marginal zone and the ventricular zone, a third area form which is called intermediate zone or mental zone. So when the cells in this area divide, they initially form the neuroblast cells, and these neuroblast cells then give rise to the neurons and all of the nervous tissue of the brain and the spinal cord. When this ventricular zone, cells in the ventricular zone ceases to form the neuroblast, then they will start forming the glioblast, and glioblast will form the macroglia, which will include all of the supporting cells of the central nervous system, and the remaining cells in the margin. And here is the marginal zone, where you can see axons of these neurons are running through this. This will form the white matter of the spinal cord, and the cells which remain over here after forming the neuroblast and the glioblast, they will form the ependymal cells. So another diagram, first we were looking at this section, now you are looking at the whole diagram, and in this diagram you can see this is the central canal. This is the canal which is formed after closure of the neural tube, and this hole is the epithelium. Initially this epithelium is the stratified one. This area is the ventricular zone, in which cells divide repeatedly mitotically, and they will migrate into the other zone. This area is intermediate zone, which is also called the mantle zone, and this area is the marginal zone, where you can see the axons which are running as well. And so because of the differential growth rate of these different areas, you can see this is called the roof plate, and this area is called the floor plate. And because of the thickening of the lateral walls, sulcus appear over here, which is called the sulcus limitans, which will divide these two areas into alar plate and the basal plate. So in this diagram you can also see this is the roof plate. This area is the central canal. Here is the floor plate, you can see. This area is the dorsolateral lamina, which will form the dorsal horn, and it is called the Eller plate. And this area is the ventral area, which will form the ventral as well as the lateral horn of the spinal cord, and outer to this is the white matter. Similarly, in this diagram you can also see this is the dorsal column of the gray matter, this one is the lateral column of the gray matter, this is the anterior column of the gray matter. Here you can see the ventral roots which are emerging, then passing through the outer zone, that is the marginal zone, which is the area which contains the white matter of the spinal cord, and they forming the ventral nerve roots. Similarly, here you can see the dorsal nerve roots also. These areas shown by different color. This is the sensory region, this is the autonomic region, and this is the motor region. So in this diagram you can also see this is the roof plate and this area is the floor plate. Here is the central canal. Here you can see the dorsal spinal nerve root ganglion. This is the spinal root ganglion which is present over here, and these are this area is the marginal zone, and this is the cells of the intermediate or mental zone, and this is the area which is called the ventricular zone. So these three zones of the neural tube are shown. Again I repeat, ventricle zone, the intermediate or the mental zone, and then the marginal zone. This is the floor plate, this is the roof plate, this is the posterior horn forming, and the anterior horn which is forming. So development of spinal cord, again you can see in this diagram, this is the central canal with the roof and the floor plate, and this outer area is marginal zone. This is the Eller plate which is a sensory, which is present in the dorsal region, and this is the basal plate which will form the anterior as well as lateral horn of the spinal cord. So in further differentiation you can see because of the differential growth rate, anteriorly ventral median fissure is formed, and posteriorly dorsal median sulcus with the dorsal septum is forming. Here you can see different cells taken through this area of the neural tube. These are the neuroepithelial cells, and these are the different areas. This is actually the ventricular zone, then the intermediate zone, and the marginal zone. Again taken through this area. So ventricular zone facing the lumen of the central canal. The intermediate zone is the area in between the two, and marginal zone is outer to which is the white matter, and then the spinal meninges are present over there. So again this is the enlarged view of the same diagram. You can see the roof plate, the floor plate, the Eller plate which is dorsal, which is sensory, the basal plate which forming the lateral as well as the anterior horn of the spinal cord. Here is the sulcus limitans which divide this area into Eller and basal plates. Here is you can see the ventral median fissure and dorsal median sulcus as well, and in this diagram you can see all the different zone of the neuroepithelial cells. So proliferation and differentiation of neuroepithelial cells produces thick walls and thin roof and floor plates. So these walls are thickened and roof and floor plates are thin out. Differential thickening of lateral walls of spinal cord, shallow groove on each side, that is the sulcus limitans. Here you can see this is the sulcus limitans which divide this Eller plate and basal plate. So this groove separates Eller plate, the dorsal side, from the basal plate, the ventral side. Cell bodies in the Eller plate form the dorsal gray column, and cell bodies in basal plate form the ventral and the lateral gray columns. So this is another diagram in this diagram section through the spinal cord. Here you can see this is the central canal where you can see the roof plate and floor plate, and here you can see dorsal root of the spinal ganglion. This is Eller plate. These are the different zone, ventricular zone, and then the basal plate which is forming from the mental zone, and outer to this is the marginal zone. Here you can see floor plate. This area is the spinal root ganglion which is attached to the dorsal horn via spinal nerves. So now we move on to the different types of the cells which are forming from the neuroepithelial cells. So this is the neural tube, and these are the neuroepithelial cells which give rise to different types of the cell. First, they will form the neuroblast. These neuroblasts are initially apolar cells. Later on, they form the bipolar neuroblast, and after the formation, the processes fuses with each other in a T-shaped manner that they form the unipolar neuroblast cells. Later on, they differentiate into the proper neurons with the cell bodies, the dendrites, and the axon. So these are the neuroblast initial first cells which are forming from the neuroepithelial cells. After the neuroepithelial cells ceases to form neuroblast, then they will form the glioblast. These glioblasts, also called the spongioblast, will again give rise to two types of the cell generations: the astroblast, that will form the protoplasmic astrocyte, and the fibrous astrocytes. Similarly, oligodendroblast will form the oligodendrocytes. After their formation, then these neuroepithelial cells will form the ependymal cells, which will line all the CSF line cavities in the central canal and ventricular areas in the brain and the spinal cord. Another important point, you must remember that the microglia cells, also included in the supporting cells of the central nervous system, are actually derived from the mesenchymal cells. They are formed in the bone marrow. They are one of the cells of the monocyte macrophage lineage. They migrate from the bone marrow and they populate the area of the central nervous system, and they have a phagocytic action. So please remember, this is a very important MCQ as well, that these microglia cells do not form from the neuroepithelial cells. It actually forms from the mesenchymal cells in the bone marrow and is one of the cell of the monocyte macrophage system. So now we move on to the positional changes of the spinal cord. Again, very important from your MCQ's point of view. So in embryo, the spinal cord extends the entire length of the vertebral canal. Because vertebral column and diameter grow more rapidly than the spinal cord, the caudal end of the spinal cord gradually comes to lie at a relatively higher level. So spinal cord in newborn terminate at L2 to L3 level, and in adults it terminate at the level of the L1. So have a look at this diagram. This is the initial, in which you can see the whole of the spinal cord is present till the end of the coccygeal vertebra. And in this phase, you can see initially it is present in 24 to 26 week embryo. It is present at the level of the S1. And later on in newborn, it is present either at the level of L2 or L3. And in adults, it is usually present at the level of the L1 vertebra, but may be present higher up at the level of T12 or sometimes lower down at the level of L3. So these are the variations at the level of the positional changes of the spinal cord. Also remember, this spinal cord is covered by the sets of the meninges. So when this spinal cord ascends and it has positional changes, the nerve roots which originate at all the levels, now they form the cauda equina. Similarly, the pyometer forming the terminal phylum, which is attached to the coccygeal vertebra from the tip of the spinal cord, also called the medullary cone, to the first coccygeal vertebra. This is called the phylum terminale, or the terminal phylum, and this one is the these are the cauda equina, which are the nerve roots which are passing through the different levels. So now we move on to the myelination of the nerve fibers. So myelin sheets surrounding nerve fiber within spinal cords are formed by the oligodendrocytes. In this diagram, you can see this is the oligodendrocyte, which sends process in a tongue-like fashion, and these processes wrap themselves around the axon and form the myelin sheath of the axon. In contrast to this, in the peripheral nervous system, Schwann cell is forming the sheath of the peripheral nerves. So these are the this is the peripheral nervous system where myelin sheath is formed by the Schwann cell. Here you can see the Schwann cell nuclei as well, and this is the oligodendrocyte. So now we move on to the neural crest cells, which are forming from the edges of the neural plate and migrate in a different way, and they have a different pathways. They forming the melanocyte, the dorsal root ganglia, then the autonomic ganglia, then forming the these are the adrenal medulla, and then they migrate around the developing GI tract, forming the enteric ganglia. So these are the different derivatives of the neural crest cells. Again, have a look at this diagram. This is neural tube, and these are the neural crest cells, which are forming the Schwann cells, the cells of adrenal medulla, the cells of the autonomic ganglia, cells of the posterior root ganglia and ganglia of the cranial nerves, and then the melanocytes. This is again another diagram showing you the neural crest cells, which are having different routes, the dorsal pathway where they populate the surface ectoderm, and the ventral pathway where they migrate through ventral area and form the different ganglia. So now this is another important diagram. This is the fate map along the neural crest of the presumptive territories which yield the ectomesenchyme, sensory, parasympathetic and sympathetic ganglia, and the neural crest derived mesenchyme in normal development. In this diagram, these all different colors are derivatives of the neural crest cell. So this light green color area is the mesoectoderm, which is present over here. These are the derivatives of the neural crest cell. Then this dark area is the ectomesenchyme, forming the parasympathetic. So you can see in this diagram, these are the parasympathetic ganglia and these. So by looking at this diagram, you can see again the parasympathetic is the craniosacral in origin. Again, you can see these are the sensory ganglia which are formed from the neural crest cells present throughout the spinal cord, and these are the dorsal root ganglia, and then the sympathetic ganglia which are shown by the dotted areas. So this is the fate map along the neural crest cells. Now we move on to the meninges. As you people all know that there are the three meninges which surrounds the brain and the spinal cord. Inner one is the pia mater, middle is the arachnoid. Collectively these two are called the pia arachnoid or the leptomeninges, having the same origin from the neural crest cell, and outer two, this is the dura mater, which arises from the mesenchyme of this area. So these leptomeninges are pia arachnoid. In between they have the arachnoid granulations as well, through which they are attached to each other, and here the subarachnoid space is also present. So these arachnoid granulation also give a clue that both of these have a same origin, that is from the neural crest cells. So this is the molecular regulation of the brain development. This is the surface ectoderm, this is neural tube, and this is the notochord. So from surface ectoderm, the bone morphogenetic protein 4 and 7 are released, which induces the neural plate to form the roof plate. Similarly, the sonic hedgehog genes in the notochord stimulates this area of the neural tube to form the basal plates. So these are different factors which are responsible for the brain development. Now we move on to the congenital anomalies of the spinal cord, and please remember, these are very important from your MCQ's point of view. So first, remember that the spinal cord, here the spinal cord is developing covered by the meninges, and these are the vertebral arches, which a vertebral column forming over it. So first one, which is mild form, this is the spina bifida occulta. In this form, you can see the vertebral arches do not fuse with each other, but the spinal cord and the meninges, they are present in their proper space. This area is covered by the skin tag, where the only a tuft of the hairs are also present. So this is the spina bifida occulta. When through this spina bifida occulta, the meninges also protrude out. then this is called the meningocele. And when along with the meninges there is protrusion of the neural tissue, that is the spinal cord, this is called the meningomyelocele. So please differentiate between all of these. In spina bifida occulta only the vertebral arches do not fuse. In meningocele only the meninges protrude. In meningomyelocele, along with the meninges, the neural tissue also protrude into this space, and this is a very worse condition in which the neural fold do not closes at all, and neural tissue is exposed to the amniotic fluid. This is called the rachis. So in this diagram, you can see, first have a look at these diagram. This is the spinal cord, and here you can see the subarachnoid space through which the meninges are protruding. This is the meningocele. In this diagram, you can see along with the meninges, the tissue from the spinal cord also protrude into this defect. This is called the meningomyelocele. And in this diagram, you can see the brain tissue, the spinal cord tissue is exposed to the exterior because of the failure of the neural folds to close, and this is called the myelocele. These are different terminologies: meningocele, meningomyelocele, or myelocele, which we are using for the spinal cord when we are referring to these anomalies in the cranial tissues. Little bit changes occur. So this is the herniated arachnoid matter in the cranial meningocele. This is the meningoencephalocele. So differentiate between two. This is meningomyelocele, where the spinal cord produces, and this is meningoencephalocele, in which along with the meninges the brain tissue is exposed, and this is anencephaly, in which cranial neuropore do not closes and brain tissue is exposed to the exterior, where corrosive effect of the amniotic fluid destroys the brain tissue. Anencephaly word is actually the misnomer. Anen mean absence. It is not the absence of the brain tissue, but it is because it is exposed to the amniotic fluid, so it is damaged. And this condition is the cranioschisis, in which all of the tube is not closed and it is exposed to the exterior. So here I complete my today's class, and the references for today's class are the Langman Medical Embryology, The Developing Human by Keith Elmore, and The Gray's Anatomy by Susan Stranding, the 40th edition. Thank you very much, dear students, for your attention for today's class. Inshallah, we will meet in the next class. Till then, Allah Hafiz.